you can take over the session.
Yeah. Thank you so much, Sagar. Good afternoon to all of you. Again, it gives me pleasure to introduce to you the speaker for this session, Dr. Prashant Kole. He is working currently as a senior principal investigator at BBRC in Sinjin, Bangalore. His core functional area is bioanalysis, and he is the Discovery Working Group co-chair, leading a team of scientists in cardiovascular research. Besides cardiovascular research, he also has extensive experience in the areas of immunology, oncology, fibrosis, metabolic disorders, etc. Prior to working at BBRC, Prashant has worked as a research fellow at Clinical and Practice Research Group at the Queen's University Belfast, Northern Ireland, UK, in pediatric pharmacokinetics, wherein he worked extensively in the area of dried blood spot methodology coupled with analytical platforms. His PhD is from BITS Pilani, where he has also served as a faculty member for a few years, guiding and mentoring a lot of students for over 60 research projects. He has over 31 peer reviewed articles, 9 invited ones, book chapters, and technology application notes, besides delivering several oral and poster presentations. He also serves in the editorial positions of several prestigious, reputed analytical journals. I've had the privilege of working with him on a key project, and on behalf of his colleagues and juniors, I can safely say that Prashant is a very passionate scientist, a perfectionist, a very transparent and inclusive leader. I think I wouldn't take much of his time, and I'll conclude this introduction and hand over the stage to you, Prashant. Over to you.
Thank you so much, Sandhya. And uh, it is quite a pleasure to see you here and knowing, of course, that you're alumni of BCP. First of all, accept my thanks to all the organizers. And it's a quite a day for me, at least last couple of days, to participate in this and follow some of the talks. My talk today is on the dried blood spots, and my talk has greatly been eased by yesterday's talk by Dr. Krishna Priya and this morning with Dr. Vaishali. Uh, a lot of coincidences. This goes back to year 1999. I was pursuing my master's at Karat, and my thesis viva was conducted by Dr. Saraf. BCP was one of the places that I wanted to be, and when I was evaluated by Dr. Saraf and was suggested to pursue my PhD. I moved on to BITS Pilani because I became a faculty. Incidentally, my thesis in MPharm was effect of piperine on pharmacokinetics of rifampicin. And this relates to this morning's talk by Dr. Vaishali. And again, very fascinating topic then. I did not stop there at my master's. I continued the topic to my PhD when I was in Pilani and continued my work on various ways the PK parameters, including the biology, can be modulated by herbs or other bioenhancers. I also had a great opportunity to work at RLL Jammu, where piperine was actually patented as a bioenhancer. So I spent almost 6 months at RRL to understand various aspects of the herbs, the type of bioenhancers that actually are extracted. And then I continued my other additional work at CDRI Lucknow. Finally, I was a faculty at Pilani, and this completes the circle to me where I'm actually speaking at BCP. So it's a privilege for me, and thank you for the invitation. Okay, I'll just project my slides for today. Are my slides visible?
Yes, sir.
Thank you. Thank you. Okay, so I'll just want to take you, again, a little recap since yesterday. We learned a lot from, Krishna Priya ma'am about how to undertake a bioanalytical method development. And again, it's the vast areas where its application is. Without an quantitative bioanalysis, none of the other fields can have a measurable impact assessment, right? You can be in the formulation development or anything, pharmacology or DMPK. It's important to really accurately and selectively estimate the angle of interest, in order to conclude that particular study. As Sandhya was mentioning, my journey with dried blood spots or microsampling started in year 2007, where when I was in Ireland, there was a clinical trial which was in preterm babies around 7 months old. And if you look at the global guidance on the medications, there is no national formularies or there is no formal guidance on the dosage that could be given to preterm neonates. And that's where across the Europe, there was an evaluation of some other additional techniques like microsampling or dried blood spots, which can be brought from a very qualitative genetic assessment to a quantitative bioanalysis. And this technique greatly helped us in order to file phase 4 applications with UK MHRA and actually conduct studies in entire pediatric population. Well, it's actually a technique. It's not limited to one field or the other. I think the scope of this can be brought in many different perspectives. So just a disclaimer, I'm with it's a corporate, so I would like to put a couple of disclaimers here just for the audience and as a part of my my corporate guidance as such. All right. Okay, so I just want to position my talk in a one particular area. So as I said, the application of the microsampling is in both preclinical research and the clinical research. In today's talk, I would like to emphasize or bring most of the focus in the preclinical research. I'll based on yesterday's talk and today's, I'll touch upon some of the applications of this technique in therapeutic drug monitoring at a later stage. But as an academic researchers, I think preclinical studies are one of the important studies that gives us some idea about any kind of objectives that you want to study. So this talk limiting to preclinical research, and that's the same area where I actually implemented my learnings from clinical studies to where I work now, that is BBRC. And I'm sharing my experiences in preclinical research in my current role. So, this particular talk is basically for the rodent species because that's where most of our animal models are, and we employ these species to study many of the DMPK aspects. So I'll take you through the use of rodent species where the exposure measurement is required in DMPK and tox studies. When the species are smaller, right, there is not much blood volume to actually collect. But we will require certain minimum volume to actually do any type of quantitative estimation. For example, I need to know the exposures. I need to have a sufficient plasma or the serum in order to develop a method and measure it. To enable sufficient volume collection, there are different protocols, and very widely used protocol is composite sampling. This is very common. I would like to again compare that with serial sampling, which is nothing but the microsampling. Well, once you do a microsampling, I'll take you through in details. How exactly it is done in rodents, and how exactly are these microsamples are to be handled. And there are two ways these microsamples can be handled. One is weight, which is a usual collection criteria, and the dried ones, which we will focus on today. Again, some tools that we can use for sample handling. This is dried matrix cards or the DBS cards, and how we can go about the operational guidance. For those of you who are new to this topic, or many of you might have known this topic, I still would like to take you through the basics of the dried blood spot analysis, how the workflow is designed, and how it differentiates from conventional approach towards the liquid matrices. In order to understand better, I have included the case study in today's discussion. Again, based on the theme drug disposition, I included a case study which will actually be very useful in today's talk. Finally, I will like to emphasize few key takeaways and just a thought to all of you where you can identify scope of this technique in your own academic research. Okay, so moving on, I'm sure all of you I mean, all of us at some stage must have used rodents in our either research understanding or as a part of our learnings, maybe through pharmacology or anything else. Just for the rodents, especially in pharmaceutical research, especially in the industry, there are various species and the strains that we use. Basically, the mouse and the rat. When it comes to mouse, there are a number of strains that are specifically either designed or are preferred to do a type of studies. Very common, the bulb C or the CD1 mouse, which is generally preferred for doing a simple pharmacokinetic studies. This is also a preferred strain to conduct general toxicity studies. Again, the BALC or the CD1 are the mouse which are effectively used to design the pharmacological models of disease to assess the efficacy of the test compounds or any other interventions. Similarly, the C57 or a DBA mouse are the preferred strains to develop the pharmacodynamic models in the area of immunology or metabolic disorders or neurobiology. None of the PD studies are, the PD studies without a PK readout are completely, an understanding. You really need to correlate the exposures to the effect. So again, these are also species which are first used in PK and then parallelly used for any PD models. These are little more disease specific, so they're also used in safety pharmacological studies. Oncology is a big research area, and this is one of the most resource consuming research theme as such. In oncology, especially the nude mouse, which are immunologically compromised, and the xenograft mouse are very commonly used. The xenograft is nothing but you can actually plant the tumor, which is isolated from either the humans or other from the species, and you can grow the tumor at the back of the nude mouse, to which you can use it as a PD model. Again, the nude mouse or a xenograft mouse are a preference for immunology, immuno-oncology kind of studies, both for PK and the safety pharmacology included. There comes the transgenic. I think all of us know again, we cannot really directly experiment on the humans, right? So if you have any hypothesis, you have some drugs that you have designed, those compounds, NCEs, need to taken through a battery of discovery and developmental studies before they are tested in humans. But as we were discussing in the morning, there are some in vitro systems. For example, if you want to study the metabolism, then you have hepatocytes. You can also have hepatocytes from humans, which are donated or specifically cryopreserved to do some in vitro studies. However, the in vitro studies many times are helpful to understand, although not always correlate in vivo. So what is the best way to handle such situation? We usually use transgenic mouse where we can have knock-in or knockouts. You have a double knock-in, double knockouts, and we use this technique specifically to something which we can, from a human gene to a transgenic mouse in order to make a humanized mouse. And this is also a very effective way of developing some PK or the PD models. And these are very preferred to study the metabolism early in the discovery. Again, these are very specifically useful for doing a DDI type of studies. And today I'll take you through one of the case studies which we were using, which we aim to use in order to do the DDI predictions. Rats common, I think we use Vista or Sprog dollar rats for general PK, general toxicity. And these are the animals which are again preferred to define or design the efficacy studies or development of pharmacodynamic models. What differentiates this? I think research is one part, but using these animals also is of resource or the cost concern. What you can find on the top of this mouse is the relative cost comparisons in terms of just the dollar value. I'm not putting any specific cost, I just gave a range. The Balsi would cost you $20 an animal, and the transgenic will cost you around $500 per animal. So, depending upon the specialty of animals, if you prefer any certain strains for your research. Again, so we have animal models, we have cost, we also have some ethical considerations when we do our research in the rodent. So all of you must have heard principles of 3R, or we always get the ethics committee approval before you actually study any of the DMPK studies, or including evaluation of your drug delivery systems in vivo. Okay. Now, very interesting, and I'll relate some of these points to yesterday's talk. There are a number of studies that we undertake in this species to understand ADME properties of the drugs already established or the new chemical entities like in drug discovery. What are some traditional ADME studies? Early PK drug disposition, it is just that a healthy animal, you can do a IV or an PO administration, and then you can take the blood samples at different time points in order to understand the disposition or the PK parameters of that compound or the drug. If it is in the mouse, of course, again, as I said, it's a very small animal. You really can't have much of a blood at a given time point. So we prefer n is equal to 9, although statistically it will be n is equal to 3. And I'll elaborate that in my next slide. If it is rat, it's simple. You can use n is equal to 3 or 6 or 9 or 12 based on the strength of the study that is required for your type of research or experiment. We also use these species to study route-dependent PK disposition. Many times we use the intraperitoneal administration, subcutaneous administration, just to know if the route of the administration is changed. Do you really see a change in the disposition of a compound of the drug? And these are designed to a very specific purpose to know or understand. And to just give an example, if you're doing, if you really want to use a bioenhancer, for example, if you give it orally along with the compound of interest, what happens? In order to understand the change in the PK, is it because of change in the absorption? It is because of the metabolism, or is it because of change in the excretion? So you can simply do two simple studies. You can give oral along with the bioenhancers, or you can just give oral bioenhancer and give the compound of interest as an intravenous. So what you're doing with this, you're completely avoiding the oral absorption, and then you are actually relying on complete IV profile in presence of any metabolizer or metabolic inhibitor. Similarly, these route-dependent PK dispositions are also used based on the ultimate aim of drug discovery or drug delivery many a times. Again, as we were discussing, Dr. Prakash also brought in, not many drugs are highly soluble and highly permeable. And there needs a significant investigations early on till to the development stage. What is the right formulation to understand the pharmacokinetics of the compound? And the formulation also aids us to understand what could be the limitations that this drug could face when it goes to development stage. So as simple as that, if I just in an early stage, I will do a solution PK. If I have an advanced form of the drug, or I would say a scaled up drug, or if I have crystalline versus amorphous drug, I could actually use the suspension PK to understand how it pans out against the solution. Similarly, you can have any other type of formulations that you want to study. Many times, based on the target, there are a number of organs where we want to know if the drug has reached. And those are typical tissue distribution studies. And usual organs are liver, brain, kidney, or you can have even heart. So if you really want to relate your plasma concentrations to the target organ is in the context of either the toxicity organ or an efficacy organ. So you can always do the tissue distribution studies, but this tissue distribution will always to be compared with the central compartment, that is the blood. You can also use these animals for studying circulating phase 1 and phase 2 metabolites, and you can design preclinical DDI studies. You can also use rat, for example, to study the role of drug transporters. You can use a pan inhibitor, see if your drug is a substrate of those. So such kind of studies can be also designed in the rodent species. Simple again, the biology efficacy studies, this also need a PK-PD correlation. And when we do the biology studies, mainly there are two sets of animals, and same is the case in the toxicity. So you have one set of animals which are actually from where you have an efficacy readout. We sometimes don't want to take a blood sample from this main PD study sample because it may actually drain it might actually affect your readout, efficacy readout. So you have usually a satellite animals through which we usually collect the blood to do the PK exposures. Again, in the preclinical stage, we also have a non-GLP exploratory tox studies. So it is just single dose or a single or 3 dose early toxicokinetic studies. These are usually a dose range finding studies. These studies help us to actually design the next stage of the toxicities that are TK exposures. So you can do a repeat dose toxicity 4 days, 14 days, or 28 days. Objective of these studies is different based on the compound and the type of thing that you want to understand. What is common in all of this is the measurement of the exposure, either the drug of interest alone or drug of interest along with its metabolite and otherwise. Am I audible? Hello?
Yes, sir. There was some disturbance from the
Okay.
audience side. It's okay.
I'll
Continue.
continue. Sure.
Yes.
Thank you. Okay. And what we need is the estimate of exposures. And these studies are there for a long time. And if you look at the guidance from FDA or many of the regulatory agencies, those guiding agencies tells you what is the target biometrics is mostly it's blood. We use plasma to measure the exposure of the drug or the metabolite because it is convenient for us and it is usually a surrogate to the blood. So if you ask me, in the drug discovery, although we do PK with plasma, ultimately when we do a human PK or human dose projections, we convert the plasma exposure to the whole blood exposure by conducting some in vitro studies such as blood to plasma ratio. So plasma is a part of blood, and when you want to understand a broader picture of the compound, how it actually get absorbed, how it actually gets transported, get distributed, it is blood that it is the blood that is the most important matrix where we want to relate those effects to. But as I said, for the generations we are using plasma as surrogate to the blood. There are a few reasons. That first is it's a gold standard. I think there is a rich history of correlating plasma concentrations to the pharmacodynamic effects. If you look at these two matrices, plasma is much more cleaner as compared to whole blood. It can be easily collected, it can be easily stored, and it is transported. And if a simple protocol is you have a cold centrifugation to actually separate the plasma from the blood. You separate that, you store it in -20, -80 degrees Celsius and transport it accordingly. For the last 4 or 5 decades, there is a significant understanding how one should use plasma in the perspective of sample preparation. So whatever we have learned in the last couple of days, there are number of ways one can actually process the plasma samples. So you have simple protein precipitation, liquid-liquid extraction, solid phase extraction. You also have a solid phase micro extraction, stir barosorptive extraction. So there is a broad knowledge available. We have a rich understanding of plasma as a chemical matrix. As plasma was preferred, there is a range of other platforms or tools that are developed over the years to handle this particular biometrics. And I'm sure in your experience in the lab or research or through your readings, you would imagine the minimum processing volume is nothing but an aliquot of plasma that you use to process for your method. And you will find not many methods are below 25 microliter as a processing volume. Uh, so it's the processing volume directly relates to the sensitivity of your method. So if you can increase or decrease, it can directly affect your sensitivity. In the recent years, with the advent or the development in the field of mass spectrometry, we have got sensitive analytical platforms which can address if there is a low processing volume or you have any other issues in the method development. Okay, just to give you an day-to-day what we do, for example, in our discovery research, it's a mouse PK, and conventionally we have a composite PK design, and for microsampling you have a serial collection or serial sampling protocol. Now, what is this in a simple way, right? Look at the table at the right-hand side top. Okay, if one has to do a PK in the mouse, we need a sufficient blood volume or a processing volume of plasma, right? Let's take 25 microliter for one-time processing. And if you and the yield of plasma from the whole blood is usually around 45 to 50%. You really cannot have more than 50% of the plasma from the whole blood. It's not easy to even collect many times if the volume of the blood is too low. So in order to get the overall PK profile, what we do, we make mouse group 1, we make 3 groups. N is equal to 3 animals, and we actually, you can see 3 different color rows. So you have sample at 0.25 hour collected from mouse 1, 2, 3. At 0.5 hour, it is mouse 4, 5, 6. And at 1 hour, it is 7, 8, 9 mouse. And the left-hand side, you can see the, just a pictorial presentation. When it comes to the next time point, we go back to the first set of animals, that is mouse 1, 2, and 3. Now, this is what we conventionally do even today, basically. And how do we collect the blood in the mouse? There are multiple methods, but in order to get a good flow and less restraint for the animal, many times we use retroorbital plexus, or we call it as ROP, which is just at the junction of the eye. There's a rich blood flow, so you pierce that with the glass capillary and collect the blood from retroorbital plexus. You can also collect from the tail vein. You can also collect from the heart. I'll just take you through some more details there. But what happens in the PK profile? Each time point is from one set of animals. And you can see from the PK graph, just the depiction here, it shows you inter-animal variability. So there's already a within animal variability, but composite design usually leads to higher variability in the PK data. And this is also this is not just the PK, it also affects in the PD studies or PD models. What is the the typical volume that we take in composite sampling is around 200 to 250 microliter of the whole blood at each time point. So we get around 100 to 110 microliter plasma. What in serial sampling we do is we only take 3 animals and we take the samples from all the PK points from a single animal, as you can see from the table at the right hand side bottom. How can this be possible? Right? There are multiple sampling sites that we can use to collect the micro volumes of the blood in the range of 25 to 30 microliters, such as from the tail or saphenous vein. So we don't disturb the eye, but we can actually take an alternative sampling site. And you can collect around 25 to 30 microliter blood and use this small amount of blood to subsequently make additional aliquots. We'll see it in further. How it can help, from the composite sampling to a serial collection, or it actually completely takes off the inter-animal variable to a great extent. Now it is only 3 animal, and whatever the data you see at different time points are from the single animal. Okay, so the micro sampling. Now I haven't told you what you can do with that 30 microliter blood that we have collected in the serial sampling. That 30 microliter of blood can be processed in different ways, which I'll come at a later stage. But this micro sampling greatly help us to comply the 3R principles. I am sure all of you are aware of this. 3R stands for reduction, refinement, and replacement. Now, in the context of bioanalysis, how these 3 are related to the microsampling. Let's go through that easily. First is reduction. As we have seen from the previous slide, in case of PK studies, we can significantly reduce the number of animals. For example, in mouse, n is equal to 9 gets down to n is equal to 3. If you're doing a toxicity study or a PD study, as I said, right, you usually keep satellite animals to collect the blood for the exposures. But being a very small volume of blood, you can actually take the blood directly from the study animals. It could be from the efficacy study or a TK study. And this is applicable to both mouse and the rat. So there is no need for any additional satellite group. And you will definitely appreciate that if we have taken the exposures from the same animals where I'm having a readout for efficacy or toxicity. So this is much more reliable than having a different sets of animals for different type of readouts. Next comes the refinement. The refinement is also in a way, how can we reduce the suffering and distress to the animals? As I give an example, the retroorbital plexus sampling that is from the eye is very painful. And we take a great amount of blood from there and it is repeated. So you have a left eye, then you have a right eye. Then you again go back to the left eye to have an additional time points. Right, so that's more suffering and distress to the animal. If you use the sites and the lesser volume, it reduces the pain and the suffering of the animal in the microsampling. Again, the typical definition of microsampling is anything which is less than 50 microliter, and anything above is not a microsampling, right? If you have to, many times in PK studies or many of the different studies, you know that we can't handle this animal in their conscious. Your mouse is small, you have a restrainer, you can restrain them, you have a tail, you can take a blood. But many times rats needs to be partially anesthetized to collect the blood, especially if the collection is directly from the heart. Because it's a moving animal, you really can't actually hold it and collect the blood. So with the microsampling, you can change the site of collection and reduce the restraint time. And it is much lesser invasive than the other traditional methods. The last R, again, many of you might question me, can you really replace it? No, we can't replace it. But very interestingly, if I am a biological scientist and I need to develop a method, and those who are in the lab must have measured the requirement of total plasma for example, method development and validation, right? So you have to do a multiple combination of sample preparation. You have to do an accuracy, precision, intraday. You need a significant amount of plasma, isn't it? Or if you're using a blood, you really require a significant amount of blood to do a method development and validation. Now, I'll come to it. As our requirement of the processing volume is less, you can sacrifice less animals to actually get a smaller volume of blood that can suffice your method development and validation, for example. And I'll give you a couple of examples as we pass through this presentation. Okay, some guidance and the references are quoted here, and I'm sure most of you are aware of this. But for the sake of microsampling and dried blood spot, I just want to take you some technical stuff, some technical details which will help us to further design or understand the workflow of the dried blood spots. Now, let's understand the species, how much total blood volume is, and how much volume is safe to collect from these rodents, which will not affect the study intention, right? If you sample more, you can actually stress the animal, and that is not the true readout because animal actually has gone through physiological stress that will not give me a right PK, TK, or a PD readout. So in case of mouse, the usual reference weight is around 80 to 40 grams, and the blood volume is around 60 ml per kg. Uh, if you look at the total blood volume in male mouse, it ranges from around 1.5 to 2.4 ml, depending upon the weight, and it is lesser in the females. How much is the safe? How much is the safe volume in a single bleed? Right? You can at a time you can have 100 to 200 microliter of the blood or zero point 0.1 to 0.2 ml of blood at a single bleed. And how much you can take in one day, for example, the guidance says one should not take more than 10% of blood volume in 24 hours. So not more than 10%. You can have 10% or less. And that again goes with the gender, if it is a male or a female. In case of rat, the average weight suitable for experiment ranges from 250 to 500 grams. The blood volume is around 55 or 54 to 70 ml per kg. Again, there is a significant difference in male and females, and you can collect up to 2.5 to 3 ml in a single bleed. For example, I'll come to the blood collection site where you can collect such a significant volume in the rat. Again, the total bleed out volume is restricted to not more than 10%. Okay, at over a period of 24 hours. What happens if you take more volume? As I said, um, if you actually take more volume, there is a distress to the animal, leads to different physiological changes in the key DMPK organs such as liver, heart, or kidney, and can affect your readout. That's why we restrict up to 10%. Coming to the blood collection sites and techniques, this is again a choice that we have. And most of our protocols or institutional ethics committee protocols needs this mention, which site and which surgical technique that is to be used while collecting the blood samples. Now, in case of mouse, there is hardly any report of any jugular vein or a carotid artery cannulation. So you can use in rats, we usually do a jugular vein cannulation or a carotid artery cannulation, and through that cannulation, we can collect the blood. In case of mouse, you can do a tail snip. Another very crude method. The tail snip is nothing but you cut a small part of a tail and collect the blood. If you want more time points, you have to just keep cutting a small length of the tail and actually collect the blood. Although it is described in the literature, not really preferred. We don't do a tail snip in the rats. We actually use tail vein to collect the blood both from mouse and rat, which is the non-surgical technique. So in case of mouse and rat, there are two very preferred site of collections, which are compliant to 3R principles. One is tail vein and the saphenous vein. Saphenous vein is also suitable to have a repeat bleeds at short intervals for serial microsampling. Okay. Although mouse and rat, it is possible to collect the blood by retroorbital. It is not really comfortable to the animals. There are some extreme sample collection techniques such as cardiac puncture or decapitation. So for example, if you have to collect the entire blood from the rat or the mouse for method development for any other purpose, we usually then go to terminal sample collection techniques such as take entire collection from the heart, you can decap and then collect the whole blood from the animal. Although, this may be necessary, although not very friendly with the animals. So this is just a guidance, and this guidance helps us to design the PK study. That PK study or a DMPK or a TK or a PD study defines the scope of how much volume of the biometrics could be available for my bioanalytical development, method development validation. Okay, there's also a very interesting observation you might have. These are rodents. As if you try to catch them, the first thing that happens is they're under stress, they micturate, right? That means if you have a repeated handling on these animals, there is always a cardiac changes and there is always a micturition or a loss of a urine when you handle them. This frequency of the handling animals in a PK study also may affect the PK or the PD readout. So it is very useful if you reduce the direct handling of the animal by your hands, as well as use sites which are more amenable for lower volumes and improve the confidence of your study to have a more correct readouts, any objective of that study. Okay, just to show you how exactly this saphenous vein microbleed is done or a tail sample is done, and how we handle a small volume of 30 microliters at a single time point from a single animal. So there's a pictorial presentation. I have given the reference here just for the sake, you can refer it. So in case of the first photograph, it is saphenous vein microbleeding technique. You can locate a saphenous vein on the rear leg. You can give a puncture, and then you have number of collection devices. You can use these collection devices, and then you can decide whether you want to go for a dried blood spot or you want to further play with the small volume of blood that you have. The second picture here shows the tail vein microbleeding technique. So again, when you use a tail vein to collect the sample, you start from the rearest part of the tail. Because if you have to collect more than 3 to 5 time points over a period of 24 hours, it is better you start from the rear end and then go towards the front end of the tail. So, and again, if you have seen, you can use a warm water. In the previous days, we used to use xylene. So apply little xylene, the tail vein just comes up, you tap it, and then insert the needle to collect the blood sample, right? So these are the usual protocols that were available. There are much more sophisticated collection accessories, I would say, that are now available, including like a butterfly needle, which can have a small cannula to collect the blood. So this is just a pictorial presentation, how we can collect the small volume of blood, how you collect it and then transfer it according to the method that you want to prefer. In the picture below here, it is the sample collection card for dry blood spots. So you take the blood and then transfer it directly onto the paper, and that's nothing but your blood spot on the paper. Okay, now it's an important microsampling is the first concept. So microsampling tells you or enables us to collect multiple time smaller volume of blood. Now, how you want to handle that small volume, you still have further options. Okay, so in the left hand side figure, I have just made 3 workflows here. Okay, and you can say, for example, I still want to use plasma as my target biometrics. Can I really get a small volume of plasma? Yes, there are a number of techniques and there are a number of publications that are now available, and they're usually called capillary plasma microsampling or CMS. All these workflows we have handled, developed, and implemented in our own setup, so I can vouch for them. So if you take maybe 30 to 50 microliter of blood from the animal, you can actually use a small capillary which is EDTA coated, right? So when you collect the blood, it doesn't coagulate inside of the small capillary. Once you collect, and nowadays you get a calibrated glass capillaries which are internally coated with EDTA, right? So it can tell you how much volume have you collected. Once you get, for example, 50 microliter, you already have EDTA in that. There is a wax plate. What you do, collect the blood up to the certain mark and then just push this bottom of that capillary into wax plate. So you have sealed now the bottom where the blood will not come out, right? As simple as that. You can directly centrifuge that glass microcapillary, which will separate your small volume of plasma and the cells. And you have a cutter. You can cut at the junction and transfer the small volume of plasma as we do conventionally. What you need to have is miniatured ways of handling these biomatrices. If you don't have means to seal the capillary and then centrifuge, what is the other way around? if you have seen, you have Eppendorf tubes of size 25 microliter or 100 microliter, very small Eppendorf tubes, right? So you collect the blood, you can use the Eppendorf tubes, which are small volume, with a pre-coated EDTA, or you can use a 5 microliter of heparin or EDTA in order to avoid the coagulation. Transfer the entire blood into that small Eppendorf tube, mix it, centrifuge it, and then you can use either 96 well plate, 300, or any other small volume plates. So microsampling can enable you a conventional plasma-based workflow as well, right? What is other workflow? If you have a 50 microliter blood and you want to have the estimation of the drug in the blood itself, simple. You can take another Eppendorf tube with water, collect this small amount of blood, transfer the entire blood to the water to make it a hemolyset, right? So the cells will burst open, you mix it, now you have a blood lysate or a hemolyset that can also be used to measure the concentrations of the compound of interest. Now the capillary plasma microsampling and hemolysate are wet methods of handling the biometrics, which is conventional, but with more sophistication of handling lower volumes, you can still do it. So microsampling doesn't restrict you to the dried blood spots. It also can help you to adapt to the previous ways of working with the biometrics. But it again needs all those things that are needed for a wet type of a workflow. What otherwise you can do, you can collect the volume and then you can spot them onto the collection cards. I will take you to the tools and then dry it. So that's why the name given dried blood spot. These collection cards are not limited to the blood. You can also use dried plasma spots, dried serum spots, dried CSF spots, or including dried urine spots, because the term is only that they have a collection paper to which you can add the matrix and dry it, right? So it's one of the matrix is blood, and that is the drying of the blood helps you in a different, altogether a different level. In yesterday's talk, we heard, right, when you collect the blood from the volunteers or from the animals, you need a refrigerated centrifuge to centrifuge and separate the plasma, right? Once you collect the plasma, you need a specialized cold storage, -20, -80. If you want to transport them to one lab to the other, you need a cold chain to be maintained, right? So these are significant resources when we handle matrices such as plasma. Okay, and all of you must have also seen some guidance in yesterday, stability studies, right? In case of traditional plasma methods, there are a number of stability studies that are actually required by many of the regulatory agencies. So you have a freeze-thaw stability, short-term stability, or a long-term stability. Why are these required? Because one can question, right? If I'm actually using plasma, I'm storing that plasma in -80 degrees Celsius. And does it really affect the stability of my analyte of interest? It does. That's the reason we have put checks in, different checks in our method development. And if you look at the plasma as a matrix, there are different components of plasma that differs from human to human. So in order to accommodate, you have n is equal to 6 blank collection. These all thing limits or put a lot of resources to work with these matrices. How the dried blood spot help us? Let's go through a few of them. The dried blood spots is not new. It was used conventionally by Guthrie and Suzy from 1963 for inborn errors of metabolism in infants. Well, if you really look at the government of India's program, this is still done in India for the last so many years, including right now. There are a number of centers where the collection cards are there. Once the infant is born, the blood is collected from the heel prick, and the infant is actually screened if he or she has any inborn errors of metabolism that could be classified as a disease. So what we have done in as a bioanalytical community, well, this was used previously to look at some genetic markers or some biomarkers in the blood in order to classify the inborn errors of metabolism. We use the same concept to bring into mainstream bioanalysis, where we can use this for quantitation of analytical interest, and that could be anything. It could be a chemical space, it could be a biomarker, it could be a genes, and I'll come some of those applications later. Right? So it's known we have repurposed the entire thing to our advantage, which can facilitate us for many things. For example, with this, we need only low volume of sampling, around 10 to 25. And if I'm using only 10 microliter to make one blood spot, I can have two dry blood spots from 25 microliter easily, including the dead volume of your pipette tip. Right. Now, what happens when you make the dried blood spot? You don't need any additional thing. Just keep at the room temperature and let it dry naturally. You can always accelerate the drying by different means, but you don't have to. Once it dries, it is just the paper, right? And then it is very simple to store and transport. You don't need any specific storage or transport requirement other than controlling the humidity of the card, the humidity around the card. So you can have a desiccator cap or a pouch and put it in the resealable bag and store your sample right into your drawer. This doesn't need any specialized cold storage. Now, this is a big advantage, right? If you have something which is very unstable or it has got a stability challenge, like prodrugs, for example. What happens is when you take a blood and you actually put it as a spot, during the drying, it actually deactivates all the enzymes that are present in the plasma. Due to deactivation of the enzymes during the drying process, your analyte is now less subjected to, right? So it actually improves the analyte stability as compared to the wet matrices, right? For example, if I had to do a prodrug PK, right, phosphate prodrug or a glycine prodrug, any type of a prodrug is a way of enhancing the PK of the drug, right? If it has less permeability, you can improve the permeability by making a prodrug of a compound. If it is high, so they have multiple objectives. Many times when the prodrug is added to the biomatrix, the enzymes, esterases, multiple enzymes in the blood or the plasma degrades it. And if you store them in the refrigerator, -20 or -80, you still have a chance during the freeze thaw that the prodrug can actually get destabilized or it will actually degrade. But in case of dried blood spot, that risk is not there because there is no need of freeze thaw. There is no cold chain, there is no need, there is all the enzymes in the blood and thus the plasma are deactivated, and that actually leads to much more improved stability for the analyte of interest. Now, many times when we handle the samples from the patients such as hepatitis or HIV, you need a really specialized lab with actually biosafety level 2, right? Because there could be hazard with the human samples. And this is especially when it's in liquid form, like especially blood or the plasma. So you have a lot of regulations in order to handle this biohazardous samples. But if you actually take the same sample, which could be an HIV patient, or it could be because of the hepatitis patient, and it's a dried blood spot, along with the drug, all the pathogens that are present in the blood are also deactivated. So it significantly reduces the biohazard, and this has been a really good advantage of the home-based sampling for dried blood spots, especially in African countries for that matter. You can find multiple literature over there. So the biohazard risk significantly goes down with drying of the blood on the cards. And again, the next, I was just speaking of it, the patient is home sampling. So those those patients who needs a regular therapeutic drug monitoring for example, epilepsy, many different drugs, different clinical conditions, they can actually do a home sampling, take a card, take the blood, dry it, seal it, add a desiccator, and put it from the post office. And this is just not this is not just an example I'm speaking. We have done this when I was in UK. So there was a home sampling. They don't have to really travel to the lab to give a blood for the therapeutic drug monitoring. And this being a low volume, you can actually now study those populations which usually cannot be accessed for any pharmaceutical research, especially as I give an example of pediatric population. That is not easy population to include into clinical trial. And if the micro sampling type of approach is used, you may actually, comply more to their ethical requirement, can extend your research in that. So two different things. Micro sampling is one way of, micro sampling is the basic that reduces the amount of the biometrics, the blood that we collect from the experiment. And there are different ways of handling micro samples. The first could be capillary plasma microsampling. You can have a hemolyzate, which are traditional wet samples. At the same time, what can be differently done? You can use small volumes, dry them up, and can be accessed at any time. So just wanted to summarize microsampling versus the dried blood spot. Okay. Okay. Now, as a researcher, it is important to understand when shall I use microsampling and when shall I use the dried blood spot, for example, right? So it's a very simple. As you have reduced this, the processing volume of that blood, or the plasma, DBS, DPS, or you can say, any type of sample that you have dried up, you need equally sensitive and selective bioanalytical method, which can actually employ that low volume. That's the basic. If that method is not feasible, then everything else in this chart stands down. Now, many of you might see then, oh, if this is going to be the case, then 90% of us doesn't have a mass spec, and then we should not even consider the microsampling or the dried blood spot as my workflows. No, the sensitivity and selectivity are plain words, right? The sensitivity needs to be put in perspective. For example, if the dose of paracetamol is 500 milligram tablet and I have to do a PK of paracetamol in the humans, right? I really don't need a typical sensitive method. Sensitive words should be used in the context, right? Sensitivity for that study or that objective could be 10 micromolar, as against an experiment where the sensitivity requirement could be 1 micromolar, right? So let's not get driven by highly sensitive. Highly sensitive is in perspective of study, not a general understanding, right? So and selectivity is important because selectivity differentiates the entire, everything around the analyte of interest. So the very first thing is sensitive and selective bioanalytical method feasible. Is it feasible with this low volume, right? And what are the considerations? You can see a box type, potential plasma yield from microsample. As I said, if I'm going to use a micro sampling, but the plasma in my as my target biometrics, do I get a consistent recovery of at least 10 microliter so that I can use that entire 10 microliter for my bioanalytical assay? As a typical processing volume, is my plasma volume required for each assay is fulfilled? Can I actually do, for example, I was running study samples today and my batch fails. Do I have a backup samples to go back and process? And finally, many times there is a technical requirement for incurred sample reanalysis in many of the clinical studies. So is microsampling going to provide me the sample for all of this? If this provides, then is my method possible with this volume? If this is not possible, it's simple. The recommendation is discuss option with the bioanalyst and consider conventional sampling. Even in conventional sampling, you still need to discuss with the bioanalyst how much volume you would require. And that's how you can go back and design your study protocol. Now, if there is a possibility that we can develop a sensitive and selective analytical method with the low volume, what is the next question? Is the sampling burden within the acceptable limits for the main study animals? I give you two examples, right? The pharmacodynamic studies or pharmacological studies and the toxicity studies. There are main animals and there are satellite animals, right? Now, for example, some of the PD studies or pharmacological studies goes on for 1 day, 1 week, 1 month. So your pharmacological model could be of 1 month, right? And you have to actually sample throughout this duration. And toxicity study, as I said, this is a 4-day study, 14-day study, 28-day study. You have 3-month study. So you have at least 2 or 3 readouts. Day 0 readout, day 4 readout, day 14 readout. So you need multiple collections from such studies. That's why the factors to be considered are length of a study, how many number of time points you need, what is the frequency of sampling, and is there a limitation with the species for collection of blood limits? Right? So this helps you to answer this sampling burden question. If it is no, then you can use microsampling from the small group of satellite animals. So, for example, if you really cannot, take off the satellite animals, but you can reduce the number of satellite animals. For example, if I'm doing a mouse TK study, I still use 9 animals, right, in the satellite group. At least I can reduce those 9 animals to 3 animals. So it is still useful to understand a microsampling perspective in such studies. The second is, if it is yes, then are there any reservations from obtaining the samples from the main study animals, as I said. So if you can directly sample from the main study animals, you have a perfect readout. So you have PK readout, you have a PD readout, you can always make a biomarker readout during these studies. And what are the considerations here? You have a reduced body weight or a toxicity or possible steady cardiovascular effects or blood or bone marrow possible target organs. So these are little specific to type of studies that one can consider to say yes or no. And if it is no, you can use microsampling from the directly main animals. So this is a tree that can help you, before you actually start working on your studies. You can have these answers from some of the preliminary studies and then go back and you design these studies with microsampling as an option. Okay, now I told you about what is microsample, how that microsample is handled, and now we are going to a workflow for dried blood spots. So I'm using blood as my central matrix, and how we can actually understand more about it. Okay, so there are in the commercial area, there are a number of sampling cards, and what example I have given is FDA DMPK A, B, and C type of a cards. Okay, I have given some more details here. For most of the DMPK studies, we use DMPK C type of a card. Okay, now what are these cards? They are nothing but like a drawing paper. The drawing papers are thick, isn't it? Not really very thick, but around 2 to 3 millimeters of thickness. But these sample collection cards are very specifically designed in order to have a smooth spread and uniform spread of blood. So this is a cellulose matrix which is having a binders, and if it is DMPK A and B, this cellulose matrix is actually treated. So you can have SLS, sodium lauryl sulfate, which is already into the card. What happens if you actually take a whole blood and add the blood to this type of treated cards? Because of the sodium lauryl sulfate or surfactants, it will lyse the cells, denature the proteins and expose all the genetic material outside. So there are a variety of cards that are currently available in the market with the objective how you want to have pre-treatment to your sample collection and spotting. But as I said, the DMPK-C card is untreated card and which is most useful for small, most of the DMPKT studies or PD studies or tox studies in that matter. Now, as you said, I think all of us have seen the blood. It's little viscous, right? And if I had to use 10 microliter of blood, right? So if I take a new tip, take 10 microliter of my blood and I transfer the blood. Do I really transfer 10 microliter? No, it's a viscous and there is always a part of a volume that gets retained on the tip, right? But if I'm using repeatedly, that loss of blood is always common between my calibration curve, for example, or my study samples. It can negate it, right? But still, that's the variability in your handling the blood volume. Is there a alternative to that? I think many of you might have heard, right? So when I'm using the micropipettes, there are two types. The first is called air displacement pipette, which is a very common, all our lab has it. Second is called positive displacement pipette. If you want to reduce the variability of handling the blood volumes, it is recommended to use positive displacement pipette instead of air displacement pipette, right? So I'm using a 10 microliter, 50 microliter, 100 microliter blood. When I'm transferring that blood, the blood that remains in the tip can be completely drained out by positive displacement pipette. Although this is not an absolute requirement, as I said, if your process is consistent for your study samples, calibration samples, and QC samples, this type of error generally gets actually worked out, right? So two things. One, what type of card you have. Second, what is the pipetting mechanism that you want to use to make your samples. Now, is it easy to make this? There are multiple factors one should be aware of when handling the whole blood, especially making those dried blood spots. And what is the matrix we're handling here? It's the whole blood, right? And the blood without anticoagulant gets immediately coagulated. I think we call it as a CTBT. CTBT is a time, clotting time, bleeding time. Even when you collect the blood, it might take a minute or two before it starts coagulation. Usually we use this to get serum out of blood. You don't use any anticoagulant. Take the blood, slant it, leave it for 15 minutes in a warm condition, maybe around 30 degrees or 37 degrees. The serum ooze out and we use the serum for our methods. But when we are handling the blood for dried blood spot, there are two scenarios. When we want to make a calibration curve, we want to have a bulk of the blood that needs to be having anticoagulant, right? And if you don't handle the blood well, it can hemolyze. I think hemolyzed blood is very simple, right? Blood you take, just shake it 10 times vigorously and then just stand it out. Once the cell settles, the plasma is reddish in color. So it is clear indication that it's a hemolyzed blood. And those of you who are into clinical bioanalysis will definitely know when we validate the method, there is also a part which will need to be addressed for hemolyzed plasma, right? Because that's the dirtier matrix than the plain plasma, right? So when handling the blood, multiple things can happen and we need to control them. So we are bringing here some quality control criteria when we prepare the dried blood spot for that matter. What are the challenges? Technique and site of collection. For example, this is especially true in animal studies, not much in the human studies, because human studies we are well versed. We have a large volume that you can play with. We know the vein. We actually can collect from the vein and clog it, right, to stop it. But when we do something like tail vein, if your needle moves maybe by a millimeter, when you sample the blood, you actually are sampling blood and the peripheral fluids in that vein, right? Now, what you are collecting is not just the blood, but the peripheral fluid, added to the blood. And that can lead to hemolysis or actually may lead to a little dilution at least of that analytes. Use of anticoagulants, analyte stabilizers, and blood mixing, right? As I said, when I was using a microcapillary coated with the EDTA, how you mix actually is very important. You can use a small epinduct tube or a capillary. The way you shake it affects the integrity of RBCs and can avoid the hemolysis. Secondly, for example, when you are handling highly unstable analytes from the blood, many a times we add stabilizers in that, right? One of the example is PMSF. PMSF is very commonly added as a stabilizer to number of bioanalytes and biomarkers, which will arrest all the enzymes and it will improve the stability of your analyte in the whole blood. So when we are using anticoagulant, analyte stabilizers, and the blood mixing, we have to be cautious to avoid the hemolysis. Matrix handling is also a cause of hemolysis. Now, yesterday we were talking about I had to make a calibration curve. Let that be a plasma, hemolyzate, or a whole blood. There are two guidances in this. When we are using organic stocks, one should not exceed 5% of organic spiking in the blood, plasma, or any other dense biometrics. So you can, if you want to make 1 microgram per ml or 1 micromolar solution of blood or plasma, the stock solution that you're using should not be exceeding 5% of organic solvent in that particular matrix. But if you have a aqueous or a buffer stock, you can actually go up to 20% of spiking because you're not going to disturb the natural biological environment of that biometrics. So there is a two independent guidance for spiking to make a calibration curve. One, if you're using organic solvent, should not exceed 5%. Ideally, I should not go more than 2.5%. Right? So you can use 2.5 microliter for 100 microliter of your plasma or the whole blood. Right? The spiking of plasma is simple. So if you can see, you have a plasma, you take organic pharmaceuticals, you spike it. At this junction of spiking, you still have a very micro precipitation. What you do? We vigorously cyclomix it so that it becomes uniform, and then I use it for my processing. Could be a serial dilution, could be for anything. Similarly, when I'm using whole blood to spike my drug to make a calibration curve, I have to be extra cautious. It's not just the plasma, but it's the RBCs which are getting exposed to the organic solvent and they will get hemolyzed. So you reduce the organic content when we make the whole blood calibration curve. You reduce it to 2%. You spike it, gently mix it, and then you can use it for serial dilution or a parallel dilution. Finally, during all of this, the blood should not clot. So these are the challenges when we handle the blood. When we make the dried blood spot, as I said, it's a simple process. You have a blood, you decide a volume, take it, and then add it to the center of the spot and you dry it. So how can we design a quality control criteria around this, right? So what are you doing? You're actually doing a fixed volume, first of all. I'm fixing it 10 microliter and I'm adding it. So one can then start observing. Once I spot 10 microliter, what is the size of my dried blood spots after drying of 2 to 4 hours, for example, right? Size in the means of diameter. You can easily use a scale or any electronic scale to measure the diameter of that spot. So that becomes, now you know, okay, the diameter of my spot is 6 millimeter or 8 millimeter or 10 millimeter. Now, as I said, the collection cards are thick and they have a different porosity. So when you add a blood, right, it can spread on the surface, but not equally from the rear side. Okay, because it's a thick and you have a small volume. So what you can do now to understand if your process is consistent and precise, you can have diameter from the front of the card and diameter from the rear of the card. This should be consistent. This will tell you that cards you are using are consistent. They are QC'd and there is no problem with the card itself, right? And for example, look at the figure at the right hand side top. right? If your blood is hemolyzed, right? When you spot that, what you see here is a centering effect, right? If you look at here, you have a center where all the cells are there and all the hemolyzed plasma is actually spreading out. This is a very typical sign of a hemolyzed blood, right? And I have given some worst thing that could happen in the blood spotting as such. So these are, we can think around these properties and design our own criteria to qualify the sample itself. Because sample integrity and sample understanding is really important when we use any additional different type of matrix. Now, this is a slide which I want to spend a little more time because then you will definitely correlate everything to a case study, right? All of us have seen from yesterday, Dr. Krishna Priya's presentation in the morning also, what is the workflow for developing a method? And what we have seen, all those development validation factors are from conventional plasma type of matrices. And what I'm saying plasma, same are the considerations for hemolyzed as well, because it's a liquid matrix, right? However, what we are dealing is the completely dried matrix. But only parameter we know is volume of blood that we have spotted. And that is the volume that we can put in perspective in all our calculations for everything, calibration to study samples for that matter. It's very important to understand further from the perspective of bioanalysis. And let's talk about sampling paper. We were talking yesterday about how to clean the plasma sample, right? So you have protein precipitation, liquid-liquid extraction, solid phase extraction, number of ways of cleaning the matrix. Now it's a sample paper, right? First of all, you have to bring this solid dried matrix to a condition where you can extract, right? You have a less chance or you have a less opportunities to explore multiple sample preparation technique, right? And whenever we use dried blood spot, so it's a blood and the cellulose matrix of that paper together it is. You just can't, take out the blood or the RBC flakes from that. It's homogeneous. So there is always a cellulose matrix when you start extracting from the dried blood spots. Now, it's if your paper, sampling paper is matrix, along with that, there are binders, there are some excipients to make that paper, right? There could be a possibility that the drug or the analyte or the metabolite of your interest can have an actually interaction with those cellulose matrix. And that can bind, that can happen anything. So why this is important to understand, this will help you to design your sample preparation, right? Now, effect of treatment of materials on sampling papers and matrix component and drug metabolite. I told you example of DMPK A and B type of card, which are pre-treated, pre-chemically treated cards. Now, for example, if your analyte of interest is actually affected by this matrix component or sampling paper components, then you will have to critically evaluate it. So that's the reason we avoid using such type of treated papers. We want a simple DMPKC card, which is untreated, will not have other than the cellulose matrix. So nothing additional, no additional factor has to be thought of. Now, very important factor, it's very interesting how the human body and the body physics is designed, right? The blood has two important parameters, and one is called hematocrit, and the second is total protein concentration. In a simple definition, what is hematocrit? It's an extent of the cells in your blood. So, for example, you take 1 ml of blood in a very tall tube, and that 1 ml of tube has a height of 10 centimeter, and you just stand it and keep it for 10 minutes. All the cells settle, right? The hematocrit is the ratio of cells to the total height, right? Higher the hematocrit or the lower the hematocrit is the sign of the disease, like an ESR that we do, right? Erythrocyte sedimentation rate. Hematocrit is, very critical to the normal flow of blood in our body. And in case of males and females, the hematocrit is between 45 to 55%, which is maintained by the body. For example, if you go out in the sun, you sweat a lot, right? You come in the home, you will need a water because you have lost excess amount of water because of sweating, and your body needs it to actually bring back to the normal, maybe some other additional physiological reasons. If there's a lot of bleeding, then also you have lost a lot of cells, and that could also affect the hematocrit. The second most important parameter of the blood is the total protein concentration. Yesterday we were talking about the albumin, globulins as a part of drug protein binding, right? The liver is the most important organ to have normal, healthy total protein in the blood. Okay, so if the hematocrit is 45, it's normal. If it is less or above, can affect the viscosity of the blood, right? If more cells, more viscous, very less cells, more fluid. Similarly, if the total protein concentration is less than normal or above than normal, affects the fluidity of the blood. Whatever affects the fluidity of the blood will affect the dried blood spot size and spread over the blood, right? So these are two important parameters, and I will also tell you more from the validation part of view how much these are important. Then finally, you have a bioanalytical factors. Okay, drug physicochemical properties like pharmacokinetic parameters, hydrophilicity, lipophilicity. I have given these factors as a most, that one should be very accustomed to or need to know more about it. And there's a very common recommendation. When the blood to plasma ratio for a drug is more than 1, dried blood spot is preferred method. What is a drug to plasma ratio? A blood to plasma ratio. If the drug actually gets into the RBCs more, then the blood to plasma ratio is high. Anti-malarials are the best example. There are a number of drugs for which the blood to plasma ratio is much higher. And then finally, this solid to liquid extraction gets highly affected based on a number of factors like selectivity, sensitivity, sample cleanup, and what else? There are two other things. As compared to the conventional bioanalytical methods, what additional validation parameters are required for the dried blood spot type of a workflow? Now, this slide will simply explain you how to work with the dried blood spots. So now you know how to make the dried blood spots. Let's see how you can actually process them. So it is the whole blood sample, calibration curve, or in vivo study sample. So you have a simpler blood, you actually spot the blood, you dry the blood, you store them, and now you can actually punch this dried part into a processing plate or Eppendorf tube. Simple. And there are a number of mechanical devices currently available which will help you to punch this disc and take it to the Eppendorf tube to further process it. Once you take, there are two types of workflows. One is called full punch and one is called partial punch. As the name suggests, the full punch is nothing but if you take the entire blood spot of that volume, it's called full punch. If you take part of that blood spot, it's called partial punch. Okay. So you take a punch of the disc, add extraction solvent, typically acetonitrile containing the internal standard. You can then vortex mix for 30 minutes. Then you can just simply centrifuge and collect the extract. You can put all the debris away, take the extract, and then you can use any of the analytical techniques that is at your disposal. You can use LC-UV or LC-MS. Okay, now I want to put that entire thing in a simple case study, right? You can use, the title is LC-MS determination of triazolam and hydroxy metabolites in mouse mouse DBS samples, application to transgenic mouse PK studies. And this is within my group, within my organization, when the group approached us, we are going to develop we are going to develop a model. We need a bioanalytical support, right? So what was the objective? The objective was to develop selective and sensitive bioanalytical method for simultaneous determination of triazolam and its metabolites, two metabolites, alpha hydroxytriazolam and 4-hydroxytriazolam in a transgenic mouse model, which is CYP3A4 humanized mouse. That means what it is, there is a mouse in which human CYP3A4 is expressed. And our group wanted to evaluate this model. If this model works for to doing an in vivo metabolism or a DDI studies as that of human in the mouse. So this is a humanized mouse, and we are developing this DDI model here. So the method that we wanted to develop was to simultaneously estimate triazolam 2 metabolites at the dose of 0.5 mg per kg in this particular case. So what you can see on this in this slide is the structures of the analyte of interest and two of the internal standards that we use. One is alprazolam for triazolam, and it's a deuterated internal standard of alpha hydroxy triazolam, which is used for estimation of the two metabolites. Okay, now as a group, when we got this request, we huddled in and let's thought, if this is the case, what should be our approach and what should be our methodological plan? Okay, so as I said, the transgenic animals are very expensive. In this case, the humanized mouse was costing us $450 per animal. And if I sacrifice one animal, I would get maximum 1 to 1.2 ml of blood. So this was not really viable for our group to develop and validate the method, because that means I will need at least 10 to 12 ml of blood to do an entire method development and validation package. If you require 10 animals, this itself is very expensive matrix, right? So what is an alternative here? I can use a surrogate biometrics to this, and the background species for humanized mouse was bulbc animal. Right, so Balsi was a mouse which has the same genetic background as that of the humanized mouse. We thought, why not to do first initial studies to compare Balsi blood and the humanized blood, and if we can devise the sample preparation which gives us consistent recovery, then I don't need to use the humanized mouse blood for any of the development. I can use Balsi blood to do all my additional studies. So this was the first objective, right? Now, if I'm proposing a surrogate biometrics, two different strains, right? One is humanized mouse, a Balbsi mouse. Is there a hematocrit difference? Because you can see at the right hand side, this paper, this particular diagram. If your hematocrit is 25% and you use 10 microliter, your spot diameter is 6.5 millimeter. When the hematocrit is 65%, you use 10 microliter of the blood, the spot diameter is 5.5. So if change in hematocrit will definitely affect the quality of my blood, red blood spots, and if I don't use the full punch, right? If I use partial punch, then 6.5 partial punch, 5.5 partial punch will have number of challenges in quantitative determination. Right. So in the bottom, I have showed you in real life how the spotting volume leads to formation of the dried blood spot. So this particular card is for 10 microliter, 15 microliter, 20 and 25. As the volume increases, there is an increase in the diameter, but not which is not a very proportional increase, right? There is a limit to which there is a spread, and after drying, how it looks like, right? So we wanted to understand if there is a difference in strain for the hematocrit as it affects the DBS parameters. What should be our spotting volume? What type of sample integrity or QC parameter we should use? What are the, and as I said, humanized Balsi strains. We have to develop a sample preparation which gives us consistent recovery for these two matrices. And then based on the requirement, we did a validation for selectivity, linearity, accuracy, precision, stability, and stability. How it looked like? We simply took the blood from the two species and did a clinical pathology readout. For humanized mouse, the hematocrit was 47%, and for bulb C, it was 45%, plus minus 2 or 3%. That gave us a confidence that even if you use the blood, this will not significantly differ in the spread on the card, right? And in order to avoid any challenges in the partial punch workflow, because even 2-3% change in hematocrit, if it affects the dried blood spots, we wanted to use the whole spot. To incorporate the whole spot, we thought let's have the spotting volume of 10 microliters. If you go up here, the 10 microliter spot, you can see the very first spot in the bottom. So it's a simple, I have a 6 millimeter punch in my lab. So I can completely punch this whole spot of 6 millimeter, and then I can use it for processing in the for the bioanalysis. I'll give you the importance of front diameter and the rear diameter and the ratio. Now you can see for biopsy and human eyes, there was no significant difference between front and the rear diameters at a given hematocrit value. So this also actually helped us to have a confidence, even if I use biopsy blood, it will not have a significant deviation as that of the humanized mouse. Of course, we had to do analytical recovery, which I'll come to the in the subsequent slide. Okay, this is the LCMS method parameter. We have 5 analytes, drug, 2 metabolites, 2 internal standards. How did we do the sample processing? We punched 6 millimeter of disc, and then we took into 96 well plate. We added 25 microliter of water. See, this is a dried disc. In order to if our solvent has to go and extract the analyte, it has to be wet. Simply, if we add a water, then it will actually open up the channels. And when you add acetonitrile, the solid paper, which is wet, can have a complete access to organic solvent for extraction. As compared to conventional plasma method, where we add internal standard to the plasma, here we are using internal standard. So internal standard is actually in the extraction solvent instead of that we spike in the plasma in other type of workflows. Here, once we added 25 microliter, we kept it for 5 minutes. We added 100 microliter of acetonitrile containing internal standard, and we optimized the time for 45 minutes at 300 RPM, which gave us a consistent recovery. And then we centrifused further at 4000 RPM and collected the supernatant or the filtrate, right? Once I have that filtrate, I injected 2 microliter into LCMS method, and then the calibration range was 1.95 to 1000 nanomolar for each of those analytes. Okay. Now, this is the chromatogram. It's an MRM chromatogram for a blank DBS sample, which is panel A, left-hand side. The DBS sample spiked with triazolam and 2 metabolites, which is the panel B in the middle. And okay, and the C is actually a chromatogram from the study. And the limit of quantification or sensitivity for the method was 2 nanomolar for all 3 of the analytes, triazolam and 2 metabolites. We could confidently estimate up to 2 nanomolar. Now, as I said, if I'm going to use biopsy blood for estimate as a surrogate to humanized blood, how are the recoveries looking like based on our extraction protocol? So this table actually summarizes recovery at 4 different QC levels for 3 analytes for biopsy and humanized. If you carefully look at it, the extraction recoveries were good. They ranged between 85 to 90%, and they were consistent. In the bracket, I have given percentage CV. In all of these cases, the percentage CV was less than 15%. That gives me a lot of confidence on my sample preparation. If I wet the sample, I add my acetonitrile, I actually shake it for 45 minutes, centrifuge, and I will get consistent recovery. And that will definitely help me in accompanying or achieving accuracies and precision. So this table shows you intra and interday accuracies and precision. If you look at the bottom 2 lines, the overall precision was less than 10% across the QCs. The overall accuracy was plus minus 15% across, right? So this was very consistently performing method, and my I could use biopsy blood for estimating the concentrations in the humanized blood. Okay, now there are different ways you can design the stability studies based on the intent of your method, as we discussed, right? In this case, we are in a preclinical and discovery setup. We don't want to, this method was not intended for a long time. So what we did, we only calculated once the effort of the DDI model development starts, it only lasts for a month, right? So we will do a dried blood stability studies over only 30 days and at 4 QCs, because that will be sufficient to qualify all our experiments as such. And then we also did processed samples, because this matrix has analytes and other components that may have come from the cart. For both of these stability conditions, we found at all levels, it is, all the values were completely within the acceptable limits of bioanalysis. Okay, so once the method was actually developed, we actually applied this to these studies in vivo. As I said, the dose was very low. It was 0.5 mg per kg, and we could report the concentrations for triazolam and 2 of its metabolites. And in the morning, we learned how to interpret metabolite to parent ratio. As I said, these animals are humanized CYP3A4, and triazolam is a model substrate which you can actually study the metabolism and formation of 2 metabolites, alpha and 4-hydroxy. Metabolite to parent ratio suggests the extent of metabolism in that particular species. Finally, just to conclude this case, what we have learned through this was the first time where we did the estimation of 2 metabolites and the parent simultaneously. We had a combined approach of serial sampling and DBS methodology. So I told you this, serial sampling is sampling low. How you handle the low volume depends on you, and we here employed the dried blood spot methodology. We also demonstrated use of surrogate biometrics in this particular approach. We use whole spot analysis, not partial spot. And with the whole spot, that is 10 microliter of blood, gave us the sensitivity of 2 nanomolar, which is significant sensitivity, and it gave us the entire coverage of PK profile. Finally, during the sample preparation trials, the pre-addition of 25 microliter of water was a determining step. There are a number of publications where you might find the DBS punch is directly exposed to the organic solvent for the extraction. But we also did that, but addition of water actually improved the precision of the extraction. So the water addition helped us to achieve the precision of recovery. So I think that was a good learning. Finally, you can see this was the first report for in vitro blood to plasma partitioning of triazolam. That data is not here, but this was useful. Now, what are some of the key takeaways from microsampling and the dried blood spot? It actually make it feasible. It is possible that we can develop sensitive bioanalytical method with the smaller volumes of biomatrices. The microsampling or the dried matrix sample helps us multiple ways, like 3R compliance. It reduces the cost of the study. There's a very simple, it's very simple to store the samples. It doesn't need any specific additional requirement. It also provides additional analytical stability, as we mentioned. However, when we design or when we discuss the objective of the method, please define your QC parameters for your study and for the DBS as such. As compared to the plasma, you may have to do additional method validation, and this particular methodology can be from early discovery to FIH to even phase 4 clinical trials. And many companies, including Bristol Myers Squibb, for which we work, we have taken discovery program all through to the phase 1, phase 2, phase 3 clinical trials. So this is more transferable across the development cycles. Finally, this is my suggestion to all of you, where you can think to utilize the microsampling and dried blood spot type of a workflow. If you're working in pharmacology, DMPK type of studies, this is very suitable, as I said, right? And even if you don't have to have dried blood spots, you can go with the normal protocol. When you collect 300 microliter of blood at each time point, you can spare 10 microliter, 3 spots, and just store it. You can process the rest of the blood, separate the plasma. You can go with your plasma workflow. How it will help? For example, if you have to revisit some of your biometrics for additional information, you can use these things. And we have very effectively used this DBS methodology as single spot analysis. So you have PK, PD, and biomarker. For example, in case of diabetes, for example, if you have a blood spot from the diabetes treated patient, you can have a drug exposure, you have a metabolite exposure, you can develop a method for glycosylated hemoglobin from DBS. That's the clean biomarker for your sugar control. So there are various ways you can have one single sample, you have multiple readouts. You can actually have different type of sample processing that you can design. For example, many of my friends and myself, I had a transdermal drug delivery system that was intended to deliver the drug for 3 months. And we did a rat study in UK employing dried blood spot method. So you can use the microsampling approach to evaluate a long-term PK studies. And as I said, handling of animal induces stress and can affect the performance of the novel drug delivery systems. You have a less you if you have a methods to reduce the handling of animals, you have much more reliable results in your studies. The dried blood spot is suitable for wide range of chemical space. So we use it for small molecules, mila or cyclic peptides. We also have employed monoclonal. We have a LCMS methods that are specifically developed for quantitative estimation of MABs, like trastuzumab for that matter. So you just need to think how you want to take the analyte of interest out of that blood spot, and many things can be worked around it. Very suitable for therapeutic drug monitoring. As I said, there are a number of reports again, and we have practically demonstrated for epilepsy patient, and it is very useful. So if your method is validated, you know which are the factors that can affect the performance of a method. You build the trust or you build more confidence through your procedures. This can be widely used. And this can be used in both preclinical and clinical setups. Those of you who are really interested into proteomics, genomics, or metabolomics, again, dried blood spots provide a lot of flexibility. If you don't want to do it something now, you can always store this sample very simply at room temperature without a fluctuation of humidity, and you can use these samples even after 20 years. So many of these neonatal studies for genetic deformations, we use the DBS samples which are 20-year-old, had the same readout as it could be somewhere near. So many of these analytes are highly stable on this particular type of cards. With that, this is my last slide. I just want to thank everyone as a part of organizers, my colleagues, and all of you participants. Thank you.
Thank you so much, Prashant, for sharing your wealth of knowledge from DBS. And I'm sure that all the participants in this academic setup would highly benefit because they're not regularly used, as you mentioned. But knowing and learning about it from someone like you is, I think, I can see a couple of people have put up in the chat how wonderfully informative this session was. So before I come to some of the questions which were put up, I have a couple of questions, if you permit me.
Sure.
Yeah. So you mentioned something about partial punch versus total. So in what cases would you go for a partial punch?
Yeah, where you want to have some qualitative information, right? And you don't have much of the sample stored. For mostly qualitative purposes, we use the partial punch. However, having said that, there are many papers where if where even the partial punch was used in a quantitative manner. But based on the variabilities in the hematocrit, blood to plasma ratio, any parameter that can affect the quality of blood spot, I would prefer the whole punch. That will avoid all other question, change in hematocrit, disease condition, thickness, spread, all that can be avoided if we use whole punch.
All right. My second question is if we have an analyte which due to some maybe non-enzymatically having some instability issues ex vivo. So in
Right.
that case, would that be a hindrance to this type
No,
of
I
analysis?
think the sampling paper is a platform of innovation. So what we have done, I have the same DMPK paper, C paper. I have PMSF. So I took a methanolic PMSF, added into it, dried it. So I imprinted my stabilizer onto the C. I did not use A and B, right? I have a choice of stabilizer, methanolic solution, because methanol has a, acetonitrile or methanol, it will spread across, it will imprint my stabilizer, dry it, and you spot.
Okay.
So you can play with this multiple times. I think we did two type of very interesting studies. One was calibration curve by blood. So you dilute the blood as a calibration curve and spot it. Second was you spot the blank blood and actually impregnate the accelerated calibration curve.
Oh,
So you can even do that, and we have shown that both works.
that's great. So from the chat, there's 2 to 3 questions from Dr. Sneel Reddy. First, he wanted you to comment on how accurate this technique is. I think your case study kind of proves that point, but if you want to add anything more.
Yeah. I would say in the scientific world, the term is precise. Precise defines accuracy basically. If your results are consistent, they are more reliable. Accuracy is only going to the neatness, the most accurate one. And we have proved it. I mean, we have used almost 25 discovery programs in drug discovery. Other than that, I have used it for 9 clinical trials in pediatric research. And those data are accepted not only by the scientific community, including the regulatory agencies. So we have made FDA submissions with dried blood spot methods.
And he also wanted to know if this technique is useful in the current hot topic COVID-19.
Absolutely. I'll tell you where I was, we were collaborating with Waters. Okay.
Mhm.
The Waters has a dried blood spot proteomic workflow for SARS-CoV specific, identification, viral protein vector analysis.
It's interesting.
Because it reduces the biohazard, it's very simple. It is non, once it is dried, it is very easy to process in a buffer and actually do a proteolysis.
Oh, it's
And it
interesting.
is used. You will find application notes in next 3 months on dried blood spot in COVID-19.
It's great. As anybody from the audience would like to unmute themselves and maybe ask a question, please go ahead.
Prakash has a question. Can you comment on the cost of these DBS cards? Very well, it's a good, very relevant question. Each DBS card cost us $2 to $3. Not very expensive. So around 450 rupees, right? Around 400, even if you take $5 for that matter, right? It is still cheap as compared to any storage requirements, the cold storage or transportation or anything. If you buy in a bulk, maybe $2. And you have multiple vendors. We have Agilent, we have Alstrom, we have, there are 7 vendors who provides the sampling cards.
Okay. I think I missed out one question asking about how would you compare this to ELISA?
Oh, very good question. And I was bringing it to that. So what we have done, we have done this study for trastuzumab. And the monoclonal antibody. Okay. So we did a PK study of trastuzumab in mouse. Okay. And we had this dried blood spot versus plasma. And usually for biologics, ELISA type of tests are primary test. LCMS is orthogonal most of the times, right? And we have showed, and it is also in public domain, that the dried blood spot can be used for sample preparation, both for ELISA and the LCMS.
Oh.
Same extraction, two different detections.
Different techniques. Okay.
We have presented this work, and the overall DMPK parameters with dried blood spots with ELISA and LCMS were comparable over 20 days of sampling.
Let me see if there are any more questions online.
There was one question with the forensic applications.
Yes.
Well, I was in a conference 2 years ago in Europe, and there was a person from a bomb squad. Okay, and he was very keenly discussing about the dried matrix. You know where he found the application? If there is a bomb blast, there are a lot of victims, right? What you get is blood spots. If the body is burst, then you can detect even the everything in forensic, including the material used for the blast. So a very irrelevant way of expressing it, but what it can offer you is drug of abuse. In the UK, all the pubs are actually very occasionally tested for drug of abuse, right? And those are very critical samples. They are usually dried metrized and transported easily. So, there are multiple applications for in the forensic sense too.
I think it's one application with multiple uses.
Yeah, it's a concept, right? What you need to know is how can you get analytic interest out of that dried part? If you design all your sample prep, you have number of analytical techniques that can be used in combination there.
Yeah, I think that's what it's mentioned. One guest has commented on its application in doping studies.
Yes. Yeah. And nowadays, you don't have to even process or punch anything. There are two vendors who have flow through analysis through the DBS card. So what you do, you put your card, the mobile page is actually transferred through the card, and then there is a SPA cartridge after that. Okay, so what happens? The entire mobile page is flown through the DBS card. All these matrices, cellulose matrix, plasma, blood, is trapped in the column in the solid phase. There is called a trap column, and then it reverse flushes and send it directly to the mass spec. So even you don't have to punch it now. You just need to define the area through which you want to move your mobile page through, and these are all commercially available, not research limited. So you can have these solutions also, you can browse it and you will find it. So this type of innovation has led to sample collection innovation too. You have a QLEX system, QLEX, CULEX. We are also buying it. So we have 11 rats, you can QLEX and it will spot it for you. It will collect the blood, define the spotting volume, it will spot it. So it's like a disc. If not a card, it's the disc of card. It's a round disk. So it just moves, 1 time point, 2 time point, 3 time point. So it just moves around. And then you have that card ready for the punch. There's a lot of innovation that goes once the utility
of innovation, Krishna Priya, madam, you want to go ahead?
Thank you very much, sir, for this wonderful session. I had one question to ask that other than blood, is there any other matrix also used as dried?
Yes, we have used dried plasma spots. We have used dried urine
blood
spots.
and plasma. I'm talking if urine samples or other samples also we can do.
Yeah, so in our experience, we have used it for urine spots. See, what we have done, urine is best metrics for metabolomic studies. there are a lot of very hydrophilic metabolites that generated and that get excreted. So if my hypothesis is that, okay, I did a PD study and I don't have a right idea what I'm looking for as a biometric. So I use the rat urine, spot it, dry it, and keep it. Once I start understanding the biochemical changes, I can go back selectively and look at those samples for those particular analytes. We also did a bile spotting a couple of times.
Okay. Thank you. Thank you very much.
Thank you, Prashant. And if there are no more questions, I would like to hand it over to Shivsat, madam.
Yes. Thank you, Dr. Sandhya, for chairing both the sessions yesterday for Dr. Prakash and today for Dr. Prashant. It was indeed very good. I mean, Sandhya was also the right person to chair the session because I think as faculty or as participants, maybe, we are more restricted in terms of the academics and to understand more things, what are actually happening in reality into the bioanalysis lab is what we really need to understand. I still remember, when I interacted with Dr. Prashant and I said, this is the theme that, I want to conduct the FDP. So, what would be the topic that you would like to speak on? And he immediately said, my expertise is in dried blood sample analysis. And it was something which was a new term for me. So I said, yes, it's going to be something very interesting. And I said, yes, we are looking forward to hear for it. So it was really wonderful, sir, because it's absolutely something novel, something new, which, was very difficult for us even to imagine that, okay, these analysis can be done this way. So thanks a lot once again. And as a token of appreciation and your acknowledgement and contribution for this FDP, you have been attending the sessions also along with not only today's presentation. So we have a sapling which is planted on behalf of you by our students and the housekeeping staff. So I request the team to please play the video.
This is a very novel way of, I'm tempted to come and visit now. So
Yes, you must come. You must come.
Please do come. Please do come.
Beautiful. Sure, ma'am. Thank you so much.
It's a Peru sapling which has been done for you.
So sweet then.
Sagar?
Yes, I think.
Yes, ma'am, it's coming. Yeah.
So thank you once again, Dr. Prashant.
It's my pleasure, ma'am. It's so good to connect with BCP after so many years, and thank you for inviting me. I think it was really a great experience to hear both of you yesterday and today, and enhance my own understanding in many different ways. Thank you.
Dr. Sagar, you'd like to announce?
Yes, ma'am. That brings us to the end of day 3 of this faculty development program. We have some announcements to make for this faculty development program. Participants, please note that 80% attendance is compulsory to get the certificate from AICTE for this faculty development program. Okay. Along with that, we are going to have a short examination on the final day involving all the sessions, in which 60% scoring is also required. So we will be sharing all the presentations and all the materials for all the from all the speakers that we will receive. But please note that 80% attendance and 60% marks is required for you to get the certificate from AICTE. Okay, with that note, I think we can end today's session. So stay safe, stay home, and have a wonderful evening. Goodbye, everyone.
Thank you very much.
Thank
Thank
you.
you.
Thank you, Dr. Sandhya. Bye. Bye.